gluconeogenesis, so essentially the opposite of glycolysis, and this funny little side pathway that I think I've alluded to, and if not, you'll see that I kind of have when I get there, which is the pentose phosphate pathway, which is kind of like a little shunt side pathway. Again, maybe we'll get to regulation of metabolism, although now we've wasted 7 minutes. Okay. Yes?
Is this last lecture for the
Yes. Unless there's like 2 slides that we clarify everything and bring it all together. Yes. So I'm kind of hoping we can get through gluconeogenesis, pentose phosphate pathway, and then the enzymes and glycolysis that have to be regulated, which is the first part of lecture 20, which somebody's going to run up and get the slides for in 5 minutes. They're printing. Okay, so gluconeogenesis. So we've kind of talked about this already. I think we know. So that it's essentially the reverse of glycolysis, so going backwards from pyruvate to glucose. So again, why would we want to do this? So you don't have to eat Pop-Tarts. Okay, so if you have the precursors, could you go backwards to make glucose to either raise your blood sugar levels or store sugar for later? Okay, so you can store it in the form of glycogen. So again, we're going to go from small molecule precursors, maybe even lactate, go backwards all the way up to make glucose 6-phosphate to either raise blood glucose, store sugar as glycogen, or there's kind of lots of other things we might do with the sugar if we need to. Of course, we're over here. So in the liver, that kind of makes sense, right? So glycogen is stored in the liver, so it kind of makes sense if we're going to make the precursor to glycogen in the liver as well. And so we've already seen this, right? So we've seen that we have glycolysis going in that direction. And we're going to have gluconeogenesis going the other way. And how many enzymes are there in common? 7. Let's go over these kind of quickly. So we know that glycolysis has a net negative delta G. So in the way of thinking about it being energetically favorable, it is, even though that 7 of those enzymes are near equilibrium. So it probably shouldn't surprise you that it's 7, right? So 7 of the enzymes, because they're near equilibrium, that's why we can shift and do them either forwards or backwards, depending on what substrates we have available, what metabolites are around. The reactions that are irreversible are really the ones that drive the energetic favorability, so they have a very negative delta G. Okay, so we know which ones those are, right? 1, 2, 3. And what we're going to talk about later today is that those reactions serve as the control point to determine whether glycolysis is going to happen or gluconeogenesis is going to happen. Because they're so metabolically irreversible, we don't want to go down that path unless we really mean to, and so we want to keep those under tight regulation, tightly controlled. And so we're going to kind of peel back the layers of that regulation as we get going. Okay, so the important things to think about, we've talked about near equilibrium reactions depending on the levels and substrates and products, and we've talked about metabolically irreversible reactions. And so hopefully looking at the numbers might make sense, and so these are obviously made up numbers. If we have a series of reactions in a pathway and we want to keep metabolites flowing through the path, so we call that the flux through the pathway, ideally, each of those reactions should have about the same net rate. Okay, so if the net rates are not about the same, something's going to build up somewhere. Okay, and that's not ideal, because if we have these near equilibrium reactions, that means we could go backwards, which we may not want to do. So if they all have about the same net rate, that means we have a pretty steady flux through the pathway. Does that make sense? So in that sense, the net rate is the same for all of those. And so the net rate is really the forward rate minus the reverse rate. So any enzyme, we could determine what the forward rate is and what the reverse rate is. So the net is just forwards minus reverse. But what is the equilibrium constant for each of those?
Is it forward?
Right, so forward divided by reverse. Okay, so if the net rate is the same for each of these, how does the equilibrium constant differ? So let's look at 2 and 3. So K equilibrium is about 1, right? And for reaction 3, K equilibrium is about 1. What about for reaction 1? Okay, equilibrium is, no, 1,000. Okay, so we would say then that reactions 2 and 3 are near equilibrium. Reaction 1 is metabolically irreversible. Okay, so there's a very negative delta G for that one because the equilibrium constant is so high. Does that make sense? So even though the net rate is about the same as the others, there's going to be a pretty big barrier to going backwards. Does that make sense? So see how the net rate is different from the equilibrium constant. Yes? So the net rate is just forwards minus reverse. So net rate equals forward minus reverse. K equilibrium equals forward divided by reverse. Okay? So keep that in mind as we talk about some of these reactions. So that doesn't mean that the reaction is going to pile up somewhere. So that would be the net rate. So we want the net rate to be pretty steady, even if one of the reactions is metabolically irreversible. So there's a difference between flux and equilibrium versus irreversible. Okay, so let's talk about the individual reactions of gluconeogenesis. So remember, we've made it all the way through glycolysis, and we've ended with pyruvate. Okay, so let's pretend we're in a fully oxygenated system. We're not moving on to lactate. We're just thinking about pyruvate. So if we're going to go back in gluconeogenesis from pyruvate backwards to glucose, so the first bypass reaction has to get us from pyruvate to what? Phosphoenol pyruvate, right? So remember, 7 reactions are the same. The 3 reactions of glycolysis that are not shared, that are metabolically irreversible in glycolysis, because they have such a negative delta G in the glycolysis direction, we have to figure out how to get around them for gluconeogenesis. We can't get around them just by having lots of pyruvate. Okay, it's not near equilibrium. We can't just pile up pyruvate and then shift the equilibrium because there's a huge energetic barrier. We can't just reverse pyruvate kinase. Okay, so we have to figure out another way around. So it's kind of like an MC Escher picture, right? So if you have stairs going down, you have to figure out how to get stairs coming back up, but have them still be energetically downhill. Maybe that's not a good analogy. But so both reactions have to be downhill, but they have to end up in opposite places. Okay, so the reactions of gluconeogenesis that compensate for the metabolically irreversible reactions of glycolysis are called the bypass reactions. Does that make sense? So the first bypass reaction, we're going to start with pyruvate. So to get to phosphoenol pyruvate, it's actually a 2-step reaction. So we're going to introduce a new intermediate that we didn't talk about in glycolysis. So a little inconvenient, but we have to bring somebody new into the picture. So the new intermediate is called oxaloacetate. Okay, and so we're going to use a molecule of bicarbonate. So where have we seen bicarbonate before? Blood plasma, so it's the buffer in blood. So there's probably a lot of it around. Remember we talked about the ratio of bicarbonate to carbonic acid? So this is a trick question on exam 2, right? Or exam 1 maybe. So what's the ratio of bicarbonate to carbonic acid? 20 to 1. So we have a fair amount of bicarbonate around. And so we're going to activate the bicarbonate and add it to pyruvate to make oxaloacetate. So what kind of enzyme do you think does this? A ligase. Very good. Okay, so we're going to make a new carbon-carbon bond right here, right, using a molecule of ATP. Okay, so that was your clue, right? So how does this work? Okay, so this is a little bit complicated. You don't need to know the details, but what I do want you to remember is a player in the reaction called biotin. Okay, so biotin. How many of you have heard of biotin or have worked with it in a research lab? Okay, so what do you know about biotin? Okay, it has something to do with antibodies. It's kind of like an antigen. So biotin is this little group here. So that's biotin. And it's basically a vitamin. Okay, so if you look at your cereal box, it'll actually say biotin is a component of your breakfast cereal, if you eat breakfast cereal. And the protein that binds very tightly to it is called avidin. Okay, or you might also use streptavidin. Okay, so avidin is a protein that's found in birds and us, I believe. Streptavidin is the homologue found in Streptomyces. So it's a very nice lab tool. Because biotin is a small molecule, it can be attached to just about anything. So you can biotinylate a DNA molecule. You can biotinylate a drug. And then because this protein, streptavidin or avidin, binds very tightly, so it's like the tightest non-antibody interaction known in the universe, practically. So it has a KD in the 10 to the minus 15th molar. So like femtomolar dissociation constant. So basically, if you have biotin on something and you introduce avidin or streptavidin, it'll find it no matter where it is. So if you have a very small amount of something and you biotinylate it, so you attach biotin covalently to it, the protein avidin or streptavidin will find it no matter where it is. So it's a very tight interaction. So it's a good way of pulling something out of a cell or out of a reaction if you can attach biotin to it selectively. Okay, so biotin is a vitamin, not because of any kind of lab tools, but because it's used as a coenzyme in lots of reactions. Okay, so biotin is a component of coenzymes. And so this enzyme, pyruvate carboxylase, which is the bypass enzyme, gets modified at a lysine residue with a biotin. So you have a lysine side chain, and then actually I guess we should say this whole thing is biotin, shouldn't we? Okay, so there's biotin. So lysine residue gets post-translationally modified with a biotin. And what I want you to remember about the biotin coenzyme is that it's a flexible tether. Okay, and so we're going to see more than one flexible tethers by the end of the semester. And so the purpose of having this biotin, so first of all, lysine is a long side chain, right? So 4 methylene groups before the amino group. And now we have a couple more, so there's a little bit more of a chain. So essentially biotin is extending the reach of a lysine side chain so that it can grab the bicarbonate and then swing it around to another active site. Okay, so the biotin is going to get covalently attached to the carboxylate there. Okay, and so it's going to grab biotin and then deliver it to where the pyruvate is bound. Okay, so we're going to see other examples of flexible tethers that make reactions happen in more than one place.
So a tether is like an assistant. I don't know.
So have you ever played tetherball? Like the ball on the post with the string? Okay, so the rope is the tether. So it's connecting the ball to the pole. So here the lysine residue is, part of the string. I don't know. Part of the string, and then the ball is the end of the biotin, and your hand is the bicarbonate. Okay, so it can reach far. Okay, or if you're going to clean off your ceiling fans, you need like a long-handled something. So it's to extend your reach. Okay, so this tether will reach further and grab onto something and kind of pull it in. So what it's pulling in is the bicarbonate group, or the carbonate group, and then pyruvate is bound in the active site, and so then the carbonate group is delivered to the pyruvate. Yes?
Is biotin the part that you circle? Or what?
Yes.
Okay.
Yes. So this is the CO2 from the bicarbonate. Okay, so I know I'm messing up my picture here a little bit. So see the pink color of the CO2 group? So that came from the bicarbonate. So if we really wanted to be explicit about what the ATP is doing, so this doesn't draw out what the ATP is doing, but what do you think it's doing? Right. So probably the bicarbonate, right, Did I do that right? No, that's not right. Too many bonds to carbon. There we go. Okay, so let's see. OHC O minus, right? There we go. So if that's the bicarbonate, then we have the ATP, right? So A, OH, OH, OP, OP. I'm going to get too far away. Okay, so we're going to phosphorylate by doing this, right? So the carboxylate is going to grab that gamma phosphate to make this mixed anhydride, right? So that we're going to end up with So we're going to end up with that, right? And now the nitrogen of the biotin can attack there and clip that off. So it's basically activating at that carbonyl group to make that more reactive. Does that make sense?
Why can't the carbonyl attack the
Why do we need biotin? Well, so if you think about where, so let me erase all the mess here so it's a little bit more obvious. So if I draw my bicarbonate again, okay, if we're going to end up, see that carbon-nitrogen bond there? So if the nitrogen just attacked there, that's not a good leaving group. That's not going to happen. Okay, so we're basically phosphorylating at that oxygen, so now the phosphate group can leave. So basically we're making that carbon group more electrophilic, or putting the phosphate group on because it can come off more easily as a good leaving group. Yeah, so we're activating that carbonate for attack by the biotin.
But I mean, why can't the carbonate just attack the pyruvate?
Oh. Well, so I mean, I think
Will it not fit? Is that why it needs the tether?
I don't think it's that it won't fit, but still, if you look at the carbonate group, right? So if you look at the carbonate group and how it has to be, I think you're still activating that carbon. Yeah. So you're delivering the bicarbonate. So it's not to say it wouldn't diffuse. It couldn't diffuse into the active site on its own, but certainly you're increasing the. I don't want to say specificity even, but you're delivering it very intentionally. Plus, pyruvate's in the active site already. Yep. Yeah. So pyruvate's in the active site of pyruvate carboxylase, and you have to activate that carboxylate group for attack at that pyruvate. Sometimes enzymes just do what enzymes do, sadly. Okay, so what do I want you to remember about this? So key features are we're introducing a new intermediate that we didn't see in glycolysis. We're going to use this long arm of the biotinylated lysine residue to deliver the carbonate group to the pyruvate that's already bound there. And ATP is used to activate that carbonate group. Okay? So now we've made oxaloacetate. And we're still in the first bypass reaction because we haven't gotten to phosphoenolpyruvate yet. But the second half of that bypass reaction is maybe a little bit easier to consider in that, so it's a second enzyme active site basically, and we're using the phosphate group of GTP. Okay, so both of these parts of the bypass reaction, so we're going to use a whole molecule of ATP in the first step and a whole molecule of GTP in the second step. In the second step, that gamma phosphate of GTP actually ends up on the phosphoenolpyruvate. The ATP is really just to activate the reaction and make that coupled reaction go better energetically. Okay, so none of the ATP ends up in phosphoenolpyruvate. In fact, the bicarbonate CO2 doesn't even end up there. Okay, so you can see the CO2 actually leaves. So this is really just, so we're, again, because this last step of glycolysis is so metabolically irreversible, we have to kind of bend over backwards to get to phosphoenolpyruvate. So it's going to cost us a molecule of ATP and a molecule of GTP just to phosphorylate pyruvate. Does that make
Do
sense?
all of these reactions then have a slightly positive delta G?
Well, what would you think?
Yeah.
But if we couple it to ATP hydrolysis and we couple it to GTP hydrolysis, no. So they actually are favorable energetically simply because we've coupled them to ATP equivalent hydrolysis. So it's costly. You know, the whole point of metabolism is to make ATP and ATP equivalents, and now we just used up 2 of them. So it's costly in terms of our energy supply, but it also is favorably in terms of negative delta G. Okay, because we've coupled it to ATP and GTP hydrolysis. And that's kind of the point. You know, so the bypass reactions, you know, it's going to cost us something to enter gluconeogenesis, but it's also energetically favorable. So that sounds a little contradictory, but it doesn't have to be. Everybody okay with that? Okay, so that first bypass reaction is the hardest, and it's going to get even more confusing. Okay, so the weird thing is, so we've talked about all of glycolysis happening in the cytoplasm, right? So the weird thing is we're going to take a little detour and go through the mitochondria. Okay, you didn't think we were going to get to mitochondria yet, but we are. So pyruvate will enter the mitochondria. There's plenty of CO2 in the mitochondria. So the oxaloacetate is made in the mitochondria. But the weird thing is, so again, kind of inconvenient, oxaloacetate is not transported out of the mitochondria. So we make it in there, and then it's like, oh, tricked you. You can't leave. Okay, and so now we're going to do another little, it's not a bypass reaction, but it's an indirect route to deliver the oxaloacetate into the cytoplasm. So we're actually going to convert oxaloacetate to malate. So we're going to reduce the oxaloacetate to malate. Now there's a transporter to get malate out of the mitochondria into the cytoplasm, where it's going to get oxidized back to oxaloacetate to then get decarboxylated and phosphorylated to make phosphoenolpyruvate. And you're like, what on earth? Why would you want to do that? Okay, so think about the redox reactions, or the redox reaction of glycolysis. So what redox reaction did we need to use for glycolysis?
NAD plus
Okay, so what enzyme is that? It's a dehydrogenase. Very good. Glyceraldehyde 3-phosphate dehydrogenase. Right? And so did we need, which part of the redox pair did we need? Did we need the NADH or did we need the NAD+? I think we needed, oh no, we needed the NAD+, right. Make sure I gave you the right answer. Yes, we needed the NAD+, right? Because we're going to oxidize our substrate, so we needed the reduced cofactor. Okay, so therefore, if we needed the NAD plus in glycolysis, which one do you think we need for glycolysis? NADH, right? So it turns out that the NADH in the cytoplasm tends to be pretty low. So there's more NAD plus in the cytoplasm than NADH. But if we're going to do gluconeogenesis, we need NADH. So it turns out there's plenty of NADH in the mitochondria, not so much in the cytoplasm. So if we're going to enter gluconeogenesis, we need to figure out a way to boost the NADH in the cytoplasm. And so this indirect malate transporter wackiness will boost the NADH in the cytoplasm. Okay, so you see we used NADH in the mitochondria, converted it to NAD+. So now malate is basically the reduced form of oxaloacetate. We transport that out of the mitochondria, and then, it's the oxidized form of oxaloacetate. Yes, because it gets reduced, NADH gets oxidized. And so then when we reconvert it back to oxaloacetate, we are oxidizing it to make reduced NADH. So we need the NADH. So basically this malate transporter is a way of moving NADH into the cytoplasm.
malate is the oxidized form of oxaloacetate.
So malate is the
Oxidize it to get back to
let me think for a second. So NADH gets oxidized to NAD+, so oxaloacetate gets reduced to malate. So malate is reduced form of oxaloacetate. Yeah. Yeah, so if you look at the reaction in the mitochondria, NADH is getting converted to NAD+, and then outside mitochondria in the cytoplasm, NAD+ is getting reduced to NADH. So it's almost like we're moving the NADH equivalents in the mitochondria outward toward the cytoplasm.
So is this kind of like the second way to get phospholipid?
Say that again?
Is this kind of like the second way to get PEP or phospholipid?
So this is so it's the only way. So starting with pyruvate, if we want to make phosphoenolpyruvate, this is how we have to do it.
Oh, but why did we? I feel like in the bypass reaction, we skipped this.
Yes, we did. So So this is just the net reaction of what happens.
Oh, yeah.
Okay. So we're just not talking about compartmentalization. So this is, if you kind of peek behind the curtain, this is where things are happening. Okay. So the net reaction doesn't involve malate. So we're going from pyruvate to oxaloacetate to phosphoenolpyruvate, and it's just kind of like, oh, by the way, we have to go through malate. No, so it's really just redistributing things. Okay, so we're redistributing the NADH from the mitochondria into the cytoplasm so we can use it for gluconeogenesis. Okay, so malate can go back and forth freely, but the reduced cofactors can't go back and forth freely. So we're basically transporting electrons out of the mitochondria into the cytoplasm. Does that help a little bit? However, as if that wasn't complicated enough, what if we're starting with lactate? Okay, so I said we didn't care about lactate 10 minutes ago, right? But if instead of starting gluconeogenesis from pyruvate, now what if we were running away from the bear and we have excess lactate, and now we're going to do gluconeogenesis? So to go from lactate to pyruvate, what do we also get? NADH. So really, we don't need to move NADH from the mitochondria into the cytoplasm. So it turns out that there is a little bit of mitochondrial phosphoenolpyruvate carboxylase. So that's not the primary enzyme that's used, but it is used when the NADH levels in the cytoplasm are high enough that you don't need to move electrons into the cytoplasm. I can try. Again, I know that we're digging deep into, and we haven't even gotten to regulation yet. But if you think about where you're starting with your precursors to gluconeogenesis, are you starting with pyruvate or are you starting with lactate? So lactate is what compared to pyruvate? So lactate is reduced from pyruvate. Right. And so to go from lactate to pyruvate, we're going to oxidize lactate to pyruvate and reduce NAD plus to NADH. Now, look, we have NADH that we're going to need for gluconeogenesis. Okay, so we don't have to essentially move those electron equivalents from the mitochondria. There's already enough of them there.
So why is it into mitochondria at all?
Because most of the time we're starting from pyruvate, and so the NAD plus NADH balance in the cytoplasm is such that we don't have enough NADH to do gluconeogenesis.
What if you start with lactate, isn't it already in there?
Yes, but going from lactate to pyruvate is making NADH, and so that NADH can then get used for gluconeogenesis. Oh, why it goes to the mitochondria at all? I think because maybe because pyruvate carboxylase is only a mitochondrial enzyme. Yes, thank you. Everybody completely glazed over. And so this is like crazy. Sometimes we don't want to ask why biology is the way it is. It's just what we observe, right? But think about it in terms of what do we need to do gluconeogenesis? We need the pyruvate being made into phosphoenolpyruvate, and we need sufficient NADH. So if we don't have it because we've converted lactate to pyruvate, we need to kind of trick the mitochondria into letting some of the NADH out. And so this crazy, seemingly, redundant enzyme pathway is a way of moving NADH into the cytoplasm. And that's all we're going to say about that. Seems kind of weird. Okay, the second and third bypass reactions are like, don't blink, you're going to miss them. Much, much easier. Okay, which makes sense because pyruvate going to phosphoenolpyruvate, that's the committed step of gluconeogenesis. So we want to make sure we're really going to commit to this. Okay, so after that, it's a piece of cake. So the metabolically irreversible reactions of glycolysis at the beginning are the ones that use ATP to phosphorylate. Okay, so hexokinase and whatever the other kinase is, phosphofructokinase 1, right? So those are using ATP to phosphorylate a substrate. So now how do we dephosphorylate a substrate? Hydrolyze it off. Okay, very easy. So a phosphatase is going to cleave off the phosphate that a kinase put on there. So again, general theme of biology, a kinase puts a phosphate group on, a phosphatase clips it off. So pretty much anytime you find a kinase, there has to be a comparable phosphatase. Yes?
Could you not just use ADP?
So unfortunately, that would be nice. So if we used ADP, we could actually get a molecule of ATP out of it, but in fact, that doesn't happen. Okay? So the net reaction of phosphorylating and dephosphorylating doesn't get us any ATP, it just wastes ATP. So we're going to see that these enzymes have to be tightly regulated so we're not just chewing up our ATP all the time. Jessica?
I was going to ask about ADP.
Yeah, it'd be nice if it did. It doesn't. So if you look at, in fact, I think, no. Let me jump ahead to this one. Yeah, so if you look at the phosphofructokinase 1 and the fructose 1,6-bisphosphatase 1, the net reaction is just chopping up ATP. So that would not be something you'd want to do a lot of, which is why both of those enzymes are tightly regulated. Okay, so both of them, it turns out, so if you look at the net of glycolysis and the net of gluconeogenesis, both of them have negative delta Gs, so they're both energetically favorable, but only one makes ATP for us. Okay, so only in gluconeogenesis do we have a net production of ATP, only in glycolysis do we have a net production of ATP. In gluconeogenesis, we don't. We're just chopping phosphates off of things. Okay? Okay, yeah, so what's the cost of gluconeogenesis? So it's costly in terms of our building blocks for other things, but it does have a negative delta G. So it's going to take us lots of ATP, a couple of GTPs, and to reduce cofactors. So I know I skipped the bypass reaction 3. It's the same as bypass reaction 2. We're just chopping off the phosphate group. Yes, Katie?
If glycolysis and gluconeogenesis are both favorable, how is it regulated?
We'll get there. Yeah, so the 3 metabolically irreversible reactions of glycolysis and the 3 bypass reactions of gluconeogenesis have all kinds of crazy regulation. So if you thought the mitochondria was confusing, wait till we get to phosphorylation cascades and sequestering and all that craziness. Perhaps not, based on the rate that we're going. Yeah. But the weird thing is, so once you understand the regulation, it kind of makes the basic reactions make more sense. So we will definitely do it Thursday, but if we don't get to it today, it's not on the exam, but it'll just maybe help everything solidify a little bit. Okay, does that make you feel better? Yeah, so the regulation is, it's like pulling back layers of an onion. Just when you think you've mastered it, you start to cry again, right? Isn't that a good one? That was a good one. That was good. Yeah, so if we just chew up ATP, that's not ideal. Okay, we're just going to waste what we're trying to make. So that's what we're going to talk about momentarily or Thursday. So again, what affects the direction? There's a whole lot of numbers and a whole lot of things up there, but some of the reactions are controlled by substrate levels. Some are not controlled by substrate levels. What we're going to see is they're actually controlled by how much enzyme is there, and by allosteric, and by phosphorylation, and by sequestering, and by all kinds of layers of regulation that say whether we're going to go into glycolysis in that direction or gluconeogenesis in the other direction. So that's kind of the overall organism sensing what's going on in the environment. Remember, okay, so glycolysis is very important in terms of generating energy in the brain, in erythrocytes, in other tissues. I think sperm only do glycolysis, so that's important. But where does gluconeogenesis happen? Liver and kidneys. Okay, so basically you have organs that are consumers of sugar and organs that are producers of glucose for the other organs. Okay, so if your brain needs glucose because it can only do glycolysis, but your liver says, yeah, sure, I can, recycle the lactate into pyruvate, which can then be made into glucose. How does it then get to the brain? Bloodstream, right? Okay, so it goes from the liver to the brain and to everywhere else where it's needed. So we will talk about regulation, but not quite yet. Take a 2-minute stretch break. Come up and get your quiz if you want.
I was like, hydrogen It's because I write Ellie, but I write Elizabeth. That might not be why.
Yeah, I know who you are, but I can't come up with your name when you're standing in front of me. Okay. So yes.
Do you mind defining bypass again?
Oh, question about bypass again?
Yeah, what is bypass?
Bypass. So the bypass reactions of gluconeogenesis are the enzymes that are distinct in gluconeogenesis from glycolysis. Okay, so if we go back to okay, so obviously there's 7 that are shared reactions, right? So the pink ones on the left are unique to glycolysis. The blue ones on the right are unique to gluconeogenesis, and so the gluconeogenesis reactions are called the bypass reactions. So it's starting from a glycolysis-centered world and saying that in order to do gluconeogenesis, we have to bypass, go around. It's kind of like a detour from glycolysis. Okay? Okay. I can never remember how to skip slides. Okay, oops, come back. Okay, so the pentose phosphate pathway is kind of like a shunt pathway. So we've talked about gluconeogenesis, we've talked about glycolysis, so they're kind of, and I would say parallel pathways, but they're anti-parallel because they're going in opposite directions. So the pentose phosphate pathway is kind of like off in another, so it's related, but it's not in line with glycolysis or gluconeogenesis. So it's kind of like a set of side reactions that happen only when we need them to happen. Okay, and so it's oxidation of glucose, but now it's not oxidation of glucose to make pyruvate, to enter the citric acid cycle, to put electrons into the electron transport chain. It's something else that we could do with glucose 6-phosphate. Okay, so it's kind of like a shunt off glycolysis. And so we're going to make phosphates. And so when you think pentose, what might you want to think about? Well, 5, yeah. So what sugars do you know that are pentoses? Ribose and deoxyribose. Okay, those are the ones you should think about. Okay, what's fructose? It's a hexose, yeah. Okay, so ribose and deoxyribose are pentoses, aldopentoses. Of course, we know where they're mostly used, right? RNA and DNA. But how many nucleotide cofactors have we seen already? Probably more than you know, because I was cheating, and you will cheat, and just draw the business end of NADH and NAD+. But NAD, so nicotinamide adenine dinucleotide. So that means there's two nucleotides there, right? So look, there's a ribose and there's a ribose. Okay, and FAD, flavin adenine dinucleotide, also a dinucleotide, also uses ribose. We'll see more as well. Okay, so riboses are not just used in DNA and RNA, they're also used in coenzymes. Okay, so if we have sugar that we don't need to use in glycolysis, we might need it for something else. So we might need it to make cofactors that are ribose-based cofactors. We might need to use that glucose to make ribose for RNA and DNA. So under what circumstances might we need that? So rapidly dividing cells. Okay, so when do we have rapidly dividing cells? Cancer, pregnancy, hair growth, any cells that turn over rapidly, so skin, hair, epithelial lining of your digestive tract. Okay, so if you think about chemotherapy treatments, so what are the side effects of chemotherapy that you know of? Hair loss, nausea, because you're basically chewing up the inside of your digestive tract. So those cells are rapidly dividing. If you're killing them off, that's one of the things that makes you very nauseous. Okay, so you just have less protective lining of your gut. Okay, so often the chemotherapy drugs are targeting DNA and RNA biosynthesis, or some components of that. So that's why not just the tumor cells, but other rapidly dividing cells are killed. Unfortunately, cancer drugs are not as specific for cancer cells as we wish they were. They tend to target all rapidly dividing cells. So if we have rapidly dividing cells, we need more pentoses. So we may have to divert some of our glucose from glycolysis into making new cells. Okay, and so the pentose phosphate pathway is how that happens. Okay, so rapidly dividing cells are one reason we might want to do the pentose phosphate pathway. Also, we're going to see that now we're going to make a reduced cofactor, but instead of it being NADH, it's going to be NADPH. So what's the difference between them? The only difference, I made a little scribble down there, is that in NADPH, this 2' hydroxyl on this ribose is phosphorylated. Okay, and some enzymes use NADP plus, NADPH as their redox cofactor. Some use NADH, NAD plus as their cofactors. So it's kind of two separate enzyme pools that sense two different sets of redox cofactors. Okay. Say that again?
Which parts are phosphorylated?
So this hydroxyl group in NADPH is phosphorylated.
So some glucose 6-phosphate goes to gluconeogenesis and some goes to this pathway.
Well, so glucose 6-phosphate, remember, is the second metabolite of glycolysis.
Yeah.
So if we have sugar, some of it can be used in glycolysis to produce energy.
Yeah.
But if you have a tumor or if you're pregnant, not you, somebody else was pregnant, you might have to take some of your glucose, and instead of doing glycolysis, you're going to put it into this other pathway to make ribose.
Okay.
Okay.
And this is strictly for glucose, not for glucose.
Right, so we're starting with glucose. Okay, so glucose can get phosphate. So the second metabolite of glycolysis is glucose 6-phosphate. So it can then continue into glycolysis, or it can go into the pentose phosphate pathway. So we're kind of jumping back to the beginning of glycolysis again. Okay?
Yeah.
Okay. So if you're not pregnant or have cancer, you might still do the pentose phosphate pathway, but now the end product is not ribose and deoxyribose. So it's not pentoses. We're actually going to then recycle those pentoses and just make NADPH. What is NADPH used for? Fatty acids, cholesterol, and steroids. Okay, so remember I said a couple of times, humans, mammals, are not able to make sugar from fat, but we are good at making fat from sugar. Okay, so if you consume too much sugar and you have plenty of glycogen stored, then that glucose 6-phosphate could go off into this pentose phosphate pathway, not because you have cancer, but because it's like, okay, we got to get rid of all this stuff already. Let's make reduced cofactors, and now we can store fat. Okay, so it's making the tools you need to store fat, sadly. Okay, So I will tell you that I don't want you to learn all these enzymes' names. Okay, so glycolysis, you're going to know better than anything else in the world. Gluconeogenesis, there's 7 shared enzymes, and so you're going to know those pretty well. From now on, we're going to go more and more superficially throughout the rest of the semester. So especially the pentose phosphate pathway, I want you to get the big picture without knowing the enzyme names and mechanisms or anything else. Okay? So the basic overview, so on the right-hand side, we have what's called this oxidative phase. So we're going to oxidize glucose 6-phosphate. So you don't need to know what the intermediates are called. Just know that there are 2 oxidation steps so that we get 1, 2 molecules of NADPH. Okay, so glucose 6-phosphate is oxidized in 2 steps to make ribulose 5-phosphate. You don't have to know the structure of ribulose. Just know that those 2 oxidation steps result in reduction of NADP plus to NADPH. Okay, so we're going to need that NADPH for other stuff. So we're just using this pathway to make NADPH. And we're going to make ribose 5-phosphate, which can then go on and make cofactors, RNA, DNA. Okay, so that's the oxidative phase where we're going to make those reduced cofactors. If we don't need, so if we're not pregnant or cancerous, we're going to recycle that 5-carbon sugar in a whole series of just kind of recycling back and forth reactions to make more glucose 6-phosphate. Okay, so we're either going to make riboses or we're going to recycle. But in either case, we're going to make reduced cofactors, NADPH, to make that stuff. Fatty acids, steroids, cholesterol. Okay, so again, don't memorize these reactions. I want you to appreciate them, but not memorize them. Okay, so glucose 6-phosphate. Look, we made a molecule of NADPH, and we've oxidized. So see how we've oxidized at that carbon. So we've made a lactone, right? And then in the second step here, look, we make another molecule of NADPH. We actually cleave off that CO2. So remember, we're going from a 6-carbon sugar to a 5-carbon sugar. So that other carbon is lost as a molecule of CO2. And then in the second oxidative step, so we make another molecule of NADPH, and now we have another lactone made, essentially, another carbonyl group. Okay, so one oxidation step, clipping off the CO2, another oxidation step to give us basically a ribose. So both steps that make NADPH, you can see it up there, but what would their names be, probably? Dehydrogenases. Okay, so dehydrogenases are redox enzymes that use NADH or NADPH. How are electrons transferred in dehydrogenases? As hydrides. Okay, so 2 hydride transfer steps. You don't need to know the names of the enzymes except that they're dehydrogenases. You don't need to know the names or structures of the metabolites. So you should know the end products, NADPH, CO2, and ribose 5-phosphate. So that's the oxidative reactions. So if that wasn't crazy enough, so these non-oxidative reactions, so if we don't need to use the ribose, what are we going to do with the ribose with a 5-carbon sugar? Well, the net reaction is 6 times 5 carbons equals 5 times 6 carbons. Okay, so all those individual transketolase, transaldolase, transketolase, isomerase, blah, 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 is really just kind of moving the carbon groups around. So if we go through this 6 times to get 6 5-carbon sugars, 6 ribose groups, now we're going to mix them all up, rearrange them, and end up with 5 glucose 6-phosphates again. Okay, so basically we've spun things off as carbon dioxide. And so the purpose then of the pentose phosphate pathway is either to make ribose or to make reduced cofactors, or both. Okay? Questions about pentose phosphate pathway? Nope. Okay. Yes? Yeah, so that's just kind of summarizing what's on the left.
Okay.
So these are the names of them, and then that's just the accounting. Okay, so 1, 2, 3, 4, 5, 6 5-carbon sugars equals 1, 2, 3, 4, 5, 6 carbon sugars. Yep, just moving things around. Okay, and so again, you don't need to know about these. These are just the transketolase, transaldolase. However, does this cofactor sound familiar? What does TPP stand for? Thymine pyrophosphate. And what did we say it was involved in moving around? CO2 groups. Okay, so TPP is a coenzyme that's involved in carboxylation and decarboxylation reactions. Okay, so look. So that gets moved, that gets moved. Actually, yeah, essentially. Okay, so we're moving things around. Okay, oh, golly, questions?
Sorry, can you ask?
Yes.
What's the difference between
Okay, so if you think about the kinds of sugars that we talked about, so we could have a aldoketose or an aldohexose or a ketohexose. Kind of gave it away, right? So we can have aldehyde or ketone sugars, right? And so one way to convert an aldehyde to a ketone is an isomerization reaction. So like we learned about in glycolysis, right? Or a transketolase will take an aldehyde and a ketone and basically interconvert them. Okay? And so here, so this is a ketone carbon, right? And so this R1, so the blue molecules are the same kind of base molecule, and so now the ketose is converted into an aldose, and that aldose is converted into a ketose.
Do you just trade the R group then, or you trade the...
No, so you're trading the pink part, basically. Yep. Does that make sense?
Reaction to 5-carbon
to a 7-carbon. Yep. So 5-carbon. So 2 5-carbons become a 7-carbon and a 3-carbon, right? And then so transketolase. So a 7-carbon, and now transaldolase comes back to a 6-carbon. Again, big picture is glucose 6-phosphate becomes NADPH, CO2, ribose. Okay? So obviously we need to regulate that, because if glucose 6-phosphate can go into glycolysis or the pentose phosphate pathway, how are we going to figure out which one we need to do? Right? So we need kind of a traffic controller, right? And so that's basically what metabolic regulation is, figuring out which pathway we should enter. And so what does it depend on? So it depends on the amount of NADP plus in the cell. Okay, so you're going to see this little red X in a circle many times, and so that means inhibits. Okay, so NADPH is an inhibitor of the pentose phosphate pathway. That makes sense because it's an end product, so it's basically a feedback inhibition. So if NADPH levels are high, we don't really need to do the pentose phosphate pathway in order to get NADPH. So we're going to feedback inhibit. And so do you see now why it makes sense that NADPH is used instead of NADH? Okay, so they're both redox cofactors, but now we can kind of keep them, we can regulate that enzyme differently. So now the NADH levels could rise significantly without shuffling things off or without shutting down the pentose phosphate pathway. So you're regulating that pathway by something that's an end product, not of a lot of different things, but of this pathway very specifically. Okay, so they're both reduced forms of a coenzyme, but NADPH will downregulate, will feedback inhibit, but NADH won't.
So you're saying that you could continue with glycolysis?
Right. Yep. Yeah. So you can continue so if you had high levels of NADH, you would if you had high levels of NADPH, you would continue glycolysis and not go into the pentose phosphate pathway, right? Glycolysis is going to be regulated differently, but NADPH will specifically shut down pentose phosphate pathway. Okay, so it inhibits that first reaction, glucose 6-phosphate dehydrogenase. Okay. 5 minutes. What do you want to talk about for 5 minutes? Questions on... See, I will not, now that I've printed out 15 copies of the handouts for the next lecture, I'm not going to talk about regulation. Hopefully that makes you feel better about the exam. Yes? Okay. We will do regulation on Thursday, but hopefully in the context of you've been studying glycolysis and gluconeogenesis, so now it'll just serve to reinforce what you're studying. Not in an overwhelming way, but in a helpful way. So what other questions do you have that might be helpful the last 5 minutes here, or do you just want to run away and study? So the recycling reactions.
Yeah, this
That one?
I don't understand like the arrows.
You don't understand the arrows. So because, okay, that's good. So the 5 carbon sugars, ribose 5-phosphate and xylulose 5-phosphate are then both going to be substrates for that first transketolase. Okay, so because the transketolase and transaldolase are interconverting 2 sugars, they're both going to be substrates, and so kind of like a ping pong. I'm not going to say it's a ping pong mechanism, but we're bringing together 2 things. They both change to become 2 different things. Okay, so we have 2 substrates and 2 products. So if you look on the right, we don't care what the substrates and products are. It's just that we're bringing together 2 5-carbon sugars. One happens to be a ketose, one happens to be an aldose, and now they're going to come together on the enzyme. One is going to become a 7-membered aldose, and one's going to become a 3-carbon ketose. Okay, yeah, so they're both substrates. There's 2 different products, and now, yeah, they're just going to rearrange until we get 6 carbons, and then that goes off and does something else. Okay. Other questions? Okay, run off and study. I will see you Thursday. Again, I don't have office hours tomorrow because I teach 2 to 3:15 now. I will be in and around my office if you have questions. It's always safest to email. And if you walk into my office and I have a panic-stricken look on my face, I'll tell you to come back later. But feel free to stop by if you want to. Yeah. No. What's that about biotin?
Can you clarify which part is the biotin and which part is
Let me. Oh no. Let me. Yes. Biotin. Let me find the biotin. Okay, so the biotin is not the pink CO2 part. It's this fused ring. So it has that thiolactone ring or whatever it is. So it's the fused 2 5-membered rings and the chain that attaches it to lysine.
The one that you circle.
Yes, the one that I circled is biotin.
Yeah, and the pyruvate carboxylase is the one with starting with the lysine, kind of like.
Yes,
right?
so the lysine is a residue of pyruvate carboxylase.
Okay. And then, wait, without the biotin, why can't the CO2 just go off? Oh, it just helps a better living or something?
Well, so it activates the CO2.
CO2
So
on the bottom, right?
yeah,
Bottom part?
yeah. So
Okay.
this is all the same. So that CO2. So that CO2 is the same as that CO2 is the same as that CO2. You know, so it's basically the biotin serves to direct the CO2 from bicarbonate into the active site in a way that it's reactive to pyruvate.
Okay, and the CO2, you said CO2 on the top is the same as CO2.
Well, I mean, it's bicarbonate.
Okay.
So CO2 can get enzymatically converted to bicarbonate.
Yeah.
So carbonic anhydrase will convert CO2 to carbonic acid, which then is going to deprotonate to bicarbonates.
I meant like that two pinks on the top part is the same. Are the same.
Yes.
Like
So that's
that.
bicarbonate,
Yeah. That's
which
it.
then gets phosphorylated by ATP, which isn't
Yeah.
shown here. That's behind the curtains.
Yeah.
But then it gets grabbed by biotin,
Yeah.
delivered to the active site, and and then I guess it comes off of the biotin as CO2, but once it's in the active site, it's protected and it's not a problem.
Okay, cool.
Yeah, and then the pyruvate, so the electrons of that double bond, that enol,
Yeah.
so you basically have an enol, that's the enol tautomer, those electrons can grab the CO2 to make
Okay.
oxaloacetate.
Thank you.